Hello everyone, and welcome to this lesson on the introduction to immunity. As we know, our bodies are made up of cells, and these cells are rich in many different substances such as amino acids, nucleotides, electrolytes, ATP, and so on. These substances are used by the cell to carry out various essential metabolic processes such as respiration, transcription, translation, cell division, etc. In a healthy state, these cells, and hence the body as a whole, should be able to use these resources efficiently in order to carry out all the necessary processes of life. However, when our body is invaded by another organism, it can also start to use up these resources that are present in the cell. Similarly, another thing that can happen is sometimes our body develops an abnormal cell that starts using up the body's resources or releasing substances that make it harder for the healthy cells to use their resources in an efficient way to carry out the processes of life. When these kind of things happen, then that organism is no longer considered healthy. The health of that organism has been compromised. In order to ensure that an organism stays healthy and that an efficient use of its resources can be maintained, its body uses its immune system. The immune system works to prevent foreign substances from entering the body and eliminate any cells that are abnormal or any foreign substances that did manage to get through. So you could say that the job of the immune system is to defend an organism and keep it healthy. They do this by not only preventing the entry of foreign substances into the body, but also by destroying any foreign substances that do manage to get in. And they also destroy any abnormal cells that end up forming inside our body, such as cancer cells, for example. So now this raises a couple questions. Firstly, how does the immune system know which cells it should attack and which cells it shouldn't, as they're our own cells? And secondly, what are the different parts and cells that constitute the immune system? Let's start with the first of those questions. You see, each individual organism has certain membrane proteins that are going to be unique to that individual, and no other individual, with the exception of identical twins, may have those same surface proteins. So, our immune system can use these membrane proteins to recognize which cells are our own, and any material that's coming from the outside, as in a foreign organism, for example, will have different surface proteins on their membrane. Even our own cells, when they become abnormal, as we just talked about, can have different surface proteins on their membrane from normal healthy cells. So our immune system can use that to differentiate them from our own normal healthy cells. Just to remind you, our cell membrane consists of a phospholipid bilayer with proteins embedded between the phospholipids. Some of these proteins have short carbohydrate chains attached to them, and even some phospholipids have these short carbohydrate chains attached to them. When the carbohydrate chain is attached to the protein, the structure is called a glycoprotein, and on the lipid, it's called a glycolipid. Some of these glycoproteins and glycolipids that are located on the outer surface of the bilayer of the membrane are what behave as the markers for each cell. Some of these surface proteins can be used to differentiate different cell types from each other, even within the same individual. For example, the heart cell will have a different marker from a liver cell. They can also be used to differentiate the same cell type between individuals. So, for example, if you have two people, person A and person B, the markers on the liver cells of person A will be different from the markers on the liver cell of person B. So we're talking about liver cells in both cases, but because they're different individuals, the markers will be different. So these surface proteins can not only differentiate between different cell types within an individual, but also differentiate the same cell type between individuals. This ability to differentiate between the cells of one person versus the cells of another mean that our immune system has the ability to differentiate between self and non-self cells. And our immune cells are designed in such a way as to never attack self cells and to attack anything that they see as non-self. This is why, for example, after an organ transplant, in some people that tissue is rejected, even though it's the same type of tissue, such as, for example, liver tissue being replaced by other liver tissue. But because it's from another person, the immune cells see it as non-self. Even though they're both liver cells, the surface proteins on the cells of the donor will be different from the surface proteins on the cells of the recipient. And so, when the immune system sees these new surface proteins, it won't recognize them as being its own cells and will start attacking them. So, as we've seen, the immune system is using these surface proteins to determine which cells are self and which cells are non-self. This is the basis of immunity. These surface proteins are also called antigens, and they can be either self-antigens or non-self antigens. The antigens that are present on an individual's own cells are considered by the immune system to be self-antigens, and those that are not present would be non-self antigens. So if the immune cells see an antigen that it doesn't recognize as its own, it's going to attack. Now, the way our immune cells identify which cells have self-antigen and which cells have non-self-antigen is by using a particular protein on their own cell surface membrane called the Major Histocompatibility Complex, or MHC for short. Being a protein, MHC is encoded by a gene, and in order to make that protein, that gene has to first be transcribed and then translated. So the gene will first be transcribed to form mRNA, which will then be translated on the ribosome to form the primary protein structure. This structure has a very particular sequence of amino acids, which is encoded by the gene on chromosome 6. This primary protein structure is then modified in the rough endoplasmic reticulum and then transported to the Golgi complex, where it will take its final form. During this entire process, proteins acquire their secondary, tertiary, and perhaps in some cases even a quaternary structure. Once we have our final protein ready and prepared, it is then transported to the cell surface in a Golgi vesicle. Now, when it comes to genes, including all of those that code for the surface antigens or the MHC protein, for example, these genes are all inherited. During the process of sexual reproduction, the sperm from the male and the egg from the female fuse together, giving rise to an entirely new, unique cell with its very own unique genetic makeup. The specific combination of genes that this new cell has will not be shared by any other individual, with the one exception of an identical twin. Let's look at this a little closer. Now, the sperm cells and the egg cells are called gametes. So, when we say male gamete, we simply mean sperm cell, and when we say female gamete, we mean the egg cell. Now, these gametes are going to have a different combination of genes within them, even within the same individual. So, for example, this particular cell will have a different combination of genes than this one, even though they both belong to the same female individual. And same goes for the male. So this cell, for example, will have a different combination of genes than this one, even though they're coming from the same male individual. On top of this, even which gamete from each of these individuals will get the opportunity to fuse is entirely random, and each combination would give rise to a unique individual with a unique genetic makeup. Because of this, every individual person, with the exception of identical twins, has a unique genetic makeup. That means that the sequence of nucleotides in the gene will be different. This allows for the sequence of amino acids in each person's surface antigens to be unique. So, no two people, except for identical twins, will share the same structure for their antigens because the genes are different and unique. This is why our immune system is able to use these antigens to differentiate self cells from non-self cells, since each person has unique antigens. Okay, so we saw how our immune system works very hard to protect the body from invaders, as in foreign pathogens, and from any abnormal cells by recognizing the non-self surface antigens on them and killing those cells so that the individual can stay healthy. But now the question is, what makes up the immune system? It turns out the immune system consists mainly of cells and also certain chemicals like proteins which are released by those cells. Now, we can classify the immune cells into two major groups based on their function. First, we have the phagocytic cells, which include neutrophils and macrophages. These types of cells are responsible for phagocytosing or engulfing foreign cells. One thing to notice about the neutrophils is their distinct multi-lobed nuclei. This is an important feature that you can use to identify neutrophils. The second group of immune cells are the lymphocytes. These include B cells and T cells. These type of cells produce substances to kill foreign invaders. We'll talk in detail about these different cells in dedicated lessons. Now, immune cells can also be classified in another way, based on how they look under a microscope. Some immune cells have these tiny darkly staining regions or spots called granules. These cells are called granulocytes. These include the neutrophils, the basophils, and the eosinophils. The cells that don't have these granules are called agranulocytes. The prefix A indicates that they don't have the granules. These cells include the macrophages and the B and T cells, or the lymphocytes. For our lessons on immunity, we'll mainly focus on the functional classification, so the one with phagocytic cells and lymphocytes. We'll talk about each of these individual cells in more detail in other lessons. Alright, to recap, in this lesson, we discussed an introduction to immunity. We said that the job of the immune system is to keep us healthy. It does this by preventing the entry of foreign substances and destroying any foreign substances that do manage to get in, as well as destroying any abnormal cells in our body. We saw how the way our immune system differentiates between self cells and non-self cells is by using unique protein structures on our cell surface membranes called antigens. We saw how our immune cells have a special protein on their surface that helps them to identify self versus non-self antigens, called the Major Histocompatibility Complex, or MHC. Finally, we looked at the different types of immune cells and how we classify them. One way to classify them is based on their function into phagocytic cells and lymphocytes. The phagocytic cells are neutrophils and macrophages, while the lymphocytes are the B cells and the T cells. They can also be classified based on their microscopic appearance into granulocytes and agranulocytes. The granulocytes are the neutrophils, basophils, and eosinophils, while the agranulocytes are the macrophages and the two lymphocytes. And that brings us to the end of this lesson. I hope you found it helpful, and I'll see you in the next one.